Okay, so in this section what we're going to do is go under the hood. So we're going to actually go into what is the underlying part of the SMAP database. So I'm going to focus down here in terms of the underlying data within SMAP, and then Linda will take over and she'll talk about this inference engine, what it is and how it works. So you'll see that there's two components to the underlying data. We break it up. So I'll go into this first one and talk about SMAP itself. So SMAP is really mapping the spatial variability. It's identifying, as we've got here, in this parcel of land there's 2 soil types. In these other areas over here there could be up to 5 soil types mapped within what we call a polygon. So it's an area where you have a like kind of variability of soil that have been mapped together. Underneath that is a complex database. Don't expect you to read it, understand it or anything, but it's just a big database of what we call morphological attributes. So these are things we can describe in the field quickly and fast. We don't have to do a lot of detailed analysis, but it's how we describe the soil and classify the soil. So each soil type that we have mapped on SMAP has a whole database sitting behind it of key attributes. So that's what SMAP is. Underpinning that, in certain sites we have, when we've dug pits, we have taken samples, various samples, and sent away for a whole range of attributes and analysis being built up over time. So this is analytical data, stuff that's been measured in the laboratory. So and you can see there's various numbers over through there. So they're point measurements. We've gone to a dot, like this red dot here, dug a pit and characterised it fully. So that's the difference between SMAP and the NSD, but they're linked together by the inference engine to create information which we need for various decision making. The big challenges we have, we're up to 21% of the year. We've identified about 4,000 unique, what we call siblings or soil types in that area so far across New Zealand. These are soils which have different attributes of one form or the other. The National Soils Database, which I'm going to talk about in detail in a second there, only has about 1,500 profiles for the whole country. So you can see there's the mismatch between these two. We don't simply have enough analytical data for all of the range of soils that are out there within New Zealand, and that's going to grow as we add in more areas throughout New Zealand. What is the National Soils Database? It's an archival database. We inherited it from the Soil Bureau that was wound up in the early 90s. It is recognised as a nationally significant database, and there's a number of those within New Zealand, and Landcare is responsible for quite a few of those. So it is recognised as a very important database, and it's protected by statute. As I said, it's got 1,500 profiles. That sounds a lot. It sounds important, but there's a big but, and I'll show you some big buts in a second. Many of these profiles only have limited analysis. We're all interested in soil water. So soil water holding capacity, as Linda said, it's a key driver of leaching, also irrigation development, crop production and things like that. There's only 416 sites within that National Soils Database that actually have soil water data. And one point I forgot to mention before, when we said 1,500 profiles, they could be multiple profiles from one soil type. So in terms of there might only be less than 1,000 different soil types that actually have data. So once you start breaking this down, it gets worse. That's simply because data stopped being put into this database in the early 90s when we transferred from the Soil Bureau across to the CRIs. Money was pulled from actually being able to enter in new data. So what are we doing about it? One of our major drives, which we've taken off our own back, is we feel that an archival status is no longer suitable for today's needs, and there's enough evidence we've produced to do that. So we're rebuilding the database to meet modern needs, moving from archival across to alive, and that's what me and Linda will be doing once it becomes alive. We'll be quite happy. We can put data into it and make it much more useful. The other key thing is increasing its capability from classic pedology data. That's what the National Soils Database was set up for in the past, was to record data collected by pedologists. There's a lot more data being generated now around single attributes in various different ways, so we need to be much more dynamic in the database in terms of the data that we can suck in, and I'll show you some examples of that in the next few slides. So public funding in New Zealand, we generate a vast amount of soil data per annum. That's a pretty bold statement. But if we look here, this is one CRI lab. This is our environmental chemistry lab within Landcare. We have another 3 labs within Landcare. And so what do these numbers mean? Every year we're collecting around 2,700 samples. So that might be a soil layer as such, the top soil. It could be a layer in the subsoil. From those samples we're generating around 14,000 soil attributes. That could be carbon, that could be pH, that could be water holding capacity measurement, something like that. And 35% of that data comes from the non-soil scientists within our research organisation, so the ecologists, other people within the company. So it's not purely a soil scientist domain, the people that are generating soil data nowadays. So you can see, as this is over the last decade, we've generated a vast amount of data. So you just imagine what's been generated by the major soil research agencies within New Zealand every year. 3 CRIs, Plant and Food Ag Research, Landcare, universities, Lincoln, Massey, Waikato, and then there's the regional councils and ... it will go on. We're very good at generating data within New Zealand, not so good at managing it. Here's an example. These are existing national source database points in the lower part of the South Island through there, and part of stage 1 of the rebuild of the NSD is we're going to pull in around 450 profiles that we did as part of the soil survey, regional soil surveying of the lowlands in Southland there. So we're able to pull that in because it's sitting in almost a very similar database to the National Soils Database, so it's very easy to suck in. So it's our example of what we can do. So we can increase the number of sites within the NSD by 30% just this year by essentially the click of our fingers. We shouldn't say click of the fingers, it's cost a lot of money, but it shows how much data we do have out there. Another example, and I've just pulled this paper out January 2014 simply because it was of interest, so some research I'm doing at the moment. So it's research done out of Waikato University, measuring carbon and nitrogen in a paired survey of dairy and dry stock farms. So they looked at 25 paired sites, which is 50 individual sites there, 3 properties, and at 3 depths. Generates 450 attributes out of one paper. There'll be multiple other papers that have been produced by New Zealand soil scientists in that 1 month of this year, together with all the other data that's not been generated. So the key messages that I guess we want to get across here is we generate a huge amount of data per annum within New Zealand, but poor data management really means a minimal benefit for that investment. It's very hard for us all to know that what data has been generated, where it is, how it relates together, and we could all go around the room and we could probably keep going for a number of hours reminding ourselves of studies that people have done at different times, which most of us will be interested in. We're building an essential database and rebuilding the National Soils Database, but we need your support in that sense. And this slide is obviously targeted at Central Wellington as the key funders on that kind of side of things, but not only them in terms of funding, it's people taking responsibility for the data as using public money to make sure it's reused over time, so we're getting that multiplier effect. We're not getting the multiplier effect from a lot of the soil data we generate.